to the cloud. All right, and then I want to share my screen with you because I did not make a specific PowerPoint for this, but I thought I could show you some of the pictures from the book so that you could see what viruses look like, etc. So these opening pictures are just some pictures of viruses, and this is clearly, an animated drawing of a virus. That's not what a virus looks like. These are the pictures that I wanted you to see because I can't possibly draw these myself. And these are some really common viruses, and they're both the cartoon drawings of how scientists see the virus when we try to describe it to each other, as well as electron micrographs, pictures of the virus. So you can see here, this is the first virus that was ever discovered. It's called tobacco mosaic virus, or TMV. An adenovirus, pretty innocuous virus. Flu, which is very famous. Flu is an RNA virus, so you can see that it has RNA labeled here. And then bacteriophage, which is a virus we spend a lot of time talking about. And if we were going to go through all of chapter 19 in its nitty-gritty detail, I would probably spend most of my time talking to you about bacteriophage, because once you understand how bacteriophage multiplies, you can understand how pretty much any virus multiplies. One of the key important things to know about viruses, I think I just put a new slide in here, right? That's blank. Some general characteristics of viruses are as follows. So first thing you need to know is they are obligate intracellular parasites. And what we mean by that is they lack most enzymes. So basically what we're saying is they can't perform metabolism. So they can't make their own ATP, okay? And the reason for that, so thus they need a host cell. And they are fairly, we say, host cell specific. Not always, but the more closely related you are to another host, the more likely the virus can move from one species to another. But in general, plant viruses infect plants, fungal viruses infect fungus, protist virus infect protists, right? Bacteria virus infect bacteria, and animal viruses are usually relatively species specific, or at least class specific, like mammals, right? Their basic structure is they have some sort of nucleic acid or genome, right? And then they have a protein coat, always. And sometimes they have what we call an envelope. And that envelope is usually composed of a little bit of phospholipid from the host and some more proteins. And they're very tiny. Right? So we're talking about nanometers in size, in diameter. So you would need an electron microscope to actually see the virus. Now, you can see the damage they do to cells with a light microscope by studying the cells that the virus is infecting, but you can't see the virus itself unless you have an electron microscope. I'm trying to think if there's anything else that's like super key and important that I would have put on this list. That's probably it. All right. So I'm just going to leave that there and then show you these pictures of viruses. And I wanted to focus on an RNA virus because COVID-19 is an RNA virus, and this is the cycle, and your book is calling it the replicative cycle. Most of the time we refer to this as the multiplication cycle. We try not to use the word replication because that insinuates the virus is a living thing, and it technically is not a living thing. Okay, so viruses are not living things. So this virus here is sort of a generic virus. It has RNA for its genome, like our friend COVID-19. And then this is the capsid protein in purple, of course, because it's protein. And then you can see this is an enveloped virus, and it has these purple spikes sticking out of it. And the spikes are usually proteins, and those proteins are going to have receptors on the host cell. And you may say to yourself, well, why? Why does a host cell have a receptor for a virus? It's usually not on purpose. It's usually because the virus is using, it has proteins on its surface that are very similar to some protein that the cell's looking for. So when this receptor on the host cell binds to the virus protein by mistake, it tricks the host cell into bringing the whole virus in because it thinks it's something that it actually needs or wants. So remember receptor-mediated endocytosis from chapter 7. That's how the viruses oftentimes get in. Once the virus is in, we say it takes off its coat. You can see in this picture that the envelope is gone, right? And once it takes off its coat and its capsid, in this case, this virus has RNA, and this we would say is a negative or an antisense RNA. So it's the 3' to 5' strand of RNA, and it's really long. It's a lot longer than this. So once that is inside the cell, particularly inside the nucleus, the cell is going to transcribe that RNA into a copy, into messenger RNA, using the host cell's RNA polymerase. All right, so now we're going to have this messenger RNA, and we know that messenger RNA, we just talked about this yesterday, can be used for translation. So we're going to make the host cell is going to make the capsid proteins. And notice some parts of this message are going to be made into, in the ribosome, are going to be made into proteins that are going to actually transverse through the endoplasmic reticulum and the Golgi, and then they're going to be put in a vesicle. And notice they're not directly put on the virus, they're put out on the surface of your cell. Okay, so the virus is tricking your cells into making all of its key components. It's tricking your cell and using all of your cells' ATP, all of your cells' endoplasmic reticulum, all of your cells' nucleotides inside the nucleus to produce this messenger RNA that's going to produce capsid proteins, so long chains of amino acids. And it's also going to produce these glycoproteins, but notice the glycoproteins get put on the surface of your cell, of the host cell, because the virus is going to pick those up at the end. Now, viruses have two jobs that they have to complete once they get into the cell. One is to make their proteins, and the second job of equal importance is to produce more genome. So in this case, remember I said this is an antisense or a minus strand of RNA. It's going to be transcribed into a sense strand, and then that sense strand is going to be used as new template to make copies, more copies of the genome RNA that's going to get packaged up. And we call this part assembly because the virus assembles. And then this is released, and most animal viruses released not by bursting the cell open, but by what we call budding. So they come to the surface, and then they're going to collect basically some of your membrane that has the virus glycoprotein on it, and they're going to pinch off and form a new virus, and then this new virus can go on to infect neighboring cells. So I wanted to share that with you because, like I said, I think it's important for us to, it should be part of Bio 1 as well, but it's usually something that I don't get to. And in the current climate, because of the coronavirus, because we're talking about this virus all the time, I think it would be useful for you to be more informed. So I also put up this chart here of the classes of some animal viruses, and you can see the coronavirus falls under single-stranded RNA that serves as message, and these are some, and it has an envelope, and these are some of the diseases that it can cause, right? Flu virus has multiple single strands of RNA. Human immunodeficiency virus also has 2 strands of RNA. So RNA viruses are pretty common. These are some examples of DNA viruses. Papillomavirus that causes warts. Notice it's not enveloped. Herpes, which is pretty common. Type 1, type 2, as well as I think we're actually There's like 9 herpes viruses. And also that one does have an envelope, right? Ugh, no, we're not looking at that. That looks way too complex. So I wanted you to see that because, like I said, I thought it was important for you to have some idea of what this virus is doing inside of your friends and neighbors. So I'll leave that up there and I close that. And then I also will talk to you a little bit about PCR. I'm probably just going to let the HHMI video say what needs to be said, but it is a rapid method to detect nucleic acid. So in the case of the coronavirus, we're not actually detecting DNA with the polymerase chain reaction. We're actually, they call it real-time RNA or real-time RPCR because they're actually detecting RNA, not DNA. So that's what I wanted to share with you guys today. Oh, sure, Lorene, sorry. I'm kind of finished, but I will post this, so you don't have to worry. I promise that I will post this today. Questions you guys have about any of that, or any questions in general, kind of about, again, the test tomorrow, or anything that I just talked about with viruses and sort of the diagnostic testing that's going on now.
I have a question just in general about viruses. Like, what is the purpose? Is it like population control? Like why, like why does it exist?
It just does. So all organisms, all kinds of living things, if we go all, even bacteria, right? So all organisms have viruses that infect them. And the current idea is that probably there were viruses first, and we have exchanged DNA and RNA with viruses multiple times, multiple species. So if you study viruses, there are a lot of times that you'll find human genes or parts of human genes in viruses, and the same, you can find analogous genes or parts of genes in us that are probably have been there for thousands, if not millions of years, that are hanging around in our DNA as well. So since probably life on Earth started as RNA, not DNA, and RNA viruses are much more common than DNA viruses, that's kind of the thought, is that they're just a self-replicating thing, right? And clearly there are thousands of them that don't make you sick, especially all the ones that are specific for other plants or animals. And there are really, I think animal viruses have been classified into like 60 families, maybe 60 or 80 families. There's only a relatively small number that cause diseases that we've specifically identified as caused by a virus. Anything else? Anything for the test tomorrow? Anything about replication, transcription, translation, mutations, DNA structure, the people? No, nothing. You're ready? All right, then. If you have no other questions, then I will not see you tomorrow because we're having a quiz. And then Thursday is the holiday, technically. We were scheduled to have July 2nd off for the holiday. So I'm going to assign the Darwin Dangerous Idea film for you to watch over the weekend, over the long weekend. And then Monday on the 6th, when we come back, we will very quickly review chapters 22 and 23. Tuesday, we'll do 24. Wednesday, we'll do bacteria, which I forgot the number. I think it's 28, maybe, or 29. Thursday, protists. And then we'll have a day to review, for you to ask questions, and then we'll have, actually we can do a little bit of fungus. We'll do a little fungus, and then we'll have our quiz on the 15th, for our last quiz on the 15th. And then we'll be done. So if you have no other questions, then I will not see you tomorrow. I won't see you again until Monday. So good luck on the quiz, and watch the Darwin's Dangerous Idea movie. It is in lots of segments, lots of pieces on YouTube. I think it's 9 different pieces. So I will post the first one so you can find your way to find the rest of them. And then ask that you, I'm not going to post each one of those 9 segments. I don't have enough space on Canvas to do that. So you'll have to go to YouTube and fetch them out and watch them. Oh, it is? Oh, good. Thanks. I think only, Joe, so I think it's only in HOL, not HOL1. So I'll try to make sure that's fixed for tomorrow and over the weekend. All right. Then, like I said, good luck tomorrow, and I'll see you next week. Yeah, see you next week. See you Monday. All right, and the meeting. Short term.